about this really exciting, fairly recently discovered locality of bryophytes. And it's really important that we uncover these rich pre-Cenozoic bryophyte floras, because bryophytes are some of the most ancient groups of plants, but really don't understand that much about their evolution over really vast periods of geologic time, since there are relatively few diverse pre-Cenozoic bryophyte floras. And just so happens that this wonderful bryophyte flora is from Vancouver Island, which I'm sure you're all excited to hear about. First, I want to make sure everyone understands what I mean when I use the term bryophytes. The bryophytes are three different groups of non-vascular plants: the liverworts, the hornworts, and the mosses. And they actually encompass quite a lot of plant diversity today. There's more than 20,000 extant bryophyte species. Most of them are mosses, and it's very likely we're actually underestimating bryophyte diversity quite a bit. There's a lot of cryptic species that are being discovered every year. I'm going to focus on the liverworts and the mosses because that's what we have at this particular locality. There's sort of two flavors of liverworts: those that are thalloid, where their bodies are essentially flat sheets of cells, and leafy liverworts, where their bodies are aggregated into leaves and stems. I think most people are probably familiar with mosses. Mosses are, of course, like leafy liverworts. They have leaves and stems, and they're really everywhere around you on Vancouver Island. If we take a look at a moss leaf, like here on the left, you can actually see that the leaves of mosses provide a ton of taxonomically informative characters. So if we look at the leaf, there's this central midrib, which in mosses we call the costa. If you look at that midrib in cross-section, you can see a moss leaf in cross-section on the right. There's actually a lot of characters about the anatomy of that midrib that we can use to help us identify mosses. And there's just a tremendous range of variation in costal anatomy. And this is, of course, very helpful because at least the material I'll be talking about today is permineralized. So we have precisely this kind of anatomical information. The particular locality that I'll be telling you about today, we've called McEachern Bridge. And that's because this locality is in the riverbed of the Solemn River, sort of bracketing either side of where the Solemn riverbed is crossed by the McEachern Bridge near Courtney. These fossils are permineralized in calcium carbonate concretions and are considered part of the Trent River Formation, which is late Santonian to Campanian in age, so somewhere between 86 and 80 million years old. These fossils are studied in serial sections, and to produce those sections, we cut the rocks, we polish their faces, you etch the rock in very dilute hydrochloric acid, exposing just a little bit of the organic plant material. Then you melt that plant material to a sheet of cellulose acetate, some acetone. When it dries, you can peel that off, and you're left with a very thin section, something on the order of 25 to 30 microns thick, of just that plant material, which you can then study using light microscopy. And that's what all of the images I'll be showing you today are. They're those very thin sections called peels mounted on glass slides and examined in a compound microscope. Now, well, there are several concretions that contain bryophyte fossils at this McEachern Bridge locality. There's one concretion that is really exceptional. And in fact, everything I'll be showing you today comes from one roughly fist-sized concretion. And when you look at that concretion, there's just tons of bryophyte material. So if you look at the scale bar on this image, the scale bar is 50 micrometers. So your overall field of view here is less than a couple of millimeters. You can actually see 4 bryophyte shoots. Here's 1, here's 2, here's 3, and in the corner here, here's 4. And this is typical for this concretion. The bryophyte material is oftentimes tangled together by the rhizoids. The rhizoids are these little tubular structures that the bryophytes use to anchor themselves to their substratum. So things are super densely packed together, they're tangled together. And this probably means that some of these bryophytes were growing together and actually like ripped up as part of a mat of bryophyte material and eventually buried as a mat of bryophyte material together. And the preservation at this McEachern Bridge locality can be really excellent. So you have just exquisite detail of the anatomy of these plants preserved. And so in this one fist-sized concretion, there's actually 289 bryophyte shoots, which is pretty crazy. That's a sort of unheard of amount of tiny little bits of pieces of plant material in this relatively small concretion. And it's kind of interesting because this concretion isn't really anything to write home about when you look at it from the surface. You can see a fern rhizome, but it really just looks like a bunch of hashed up plant material. When you cut it and produce peels, there's just an amazing array of bryophyte diversity. And in fact, we can recognize 14 different types of bryophytes. And this makes this by far the richest pre-Cenozoic bryophyte flora that's ever been discovered. And so I'm just going to briefly walk you through 12 of these 14 types and show you the range of diversity in this one concretion. Also point out that this concretion was collected by Randall Mindel a couple of years ago. And he had the wherewithal to section this and actually recognize the crazy wealth of tiny little plants inside. First, let's take a look at the leafy liverworts. There's just one leafy liverwort morphotype. Unfortunately, I just got a heads up, my internet connection's unstable, so I apologize if I cut out here. But here you can see the morphotype in longitudinal section. Here you can see it in cross section. And it's kind of hard to interpret, in part because it's fairly simple. So if we look at the stem, there's essentially no anatomical differentiation to the stem, and the leaves are extremely delicate, not preserved all that well. But you can see they're just a single cell layer thick. And this sort of simple stem anatomy and leaves that are just a single cell layer thick is typical of leafy liverworts. It's sort of hard to push this identification any further because we don't typically study leafy liverworts in section like this. But I'm hoping as we find more specimens, as I peel this concretion, find more details and be able to identify it to a higher level. Next up are the Aquacarpus Mosses. Now, these are the mosses that are not particularly branched. If you think of mosses growing, let's say, on your roof or on a sidewalk in a little cushion, these are the Aquacarpus Mosses. And there's quite a range of Aquacarpus Mosses in this concretion. First up, we have some specimens that resemble sphagnum or peat moss. Sphagnums, of course, are a really important genus ecologically today. And we see some really unusual features in sphagnum that we can recognize in these fossils. First, if you look at the leaves, here's a section through a tiny portion of a leaf. It's actually just one cell layer thick, which is pretty unusual for a moss leaf. And there's two very different kinds of cells that make up the leaf in an alternating pattern. These tiny cells called chlorocysts, which alternate with these larger cells called leucocysts. And if we look at the stems and branches, here's a branch in cross-section, sorry, a stem in cross-section and a branch in cross-section here. The stems and branches have an inflated epidermis. So the outermost layer of cells is really inflated. That's called a hyalodermis. And so based on the occurrence of these characters, we can recognize these fossils as sphagnum, some extant sphagnum for comparison. There's actually 3 different species of sphagnum in this one concretion. And I'm going to show them to you. They all have leaves with this sort of anatomy and generally have either stems or branches with this kind of anatomy with a hyalodermis. The easiest way to distinguish these 3 species of sphagnum is actually from their stem anatomy. So I'll show you those. This is the first type. You can see the fossil on the right, similar extant anatomy on the left. This first sphagnum type just has a single cell thick hyalodermis, just found in many extant species of sphagnum. This second type, again, the fossils on the right, there's living taxa on the left for comparison, has a hyalodermis that's several cell layers thick. And this is found in several other extant species of sphagnum spread throughout many different sections of that genus. This is the third sphagnum type, and it's quite different from the others because it doesn't have, at least on its main stems, an inflated hyalodermis like that. And this is only found in one section of sphagnum section cuspidata. And these discoveries are pretty exciting because they reveal that sphagnum was quite anatomically diverse by the late Cretaceous, and critically it extends the record of extant sections of that genus back by about 25 million years. And really, this is quite a bit earlier than predicted by previous attempts to date the diversification of peat mosses using molecular techniques. It's exciting. There's another type of moss in this concretion that has leaves with 2 different cell types, those chlorocysts and leucocysts. Here we can see a leaf in cross-section with those two different cell types. It's a magnified view of that leaf. We have tiny little triangular chlorocysts and very big leucocysts. But unlike the sphagnum leaves, this leaf is several cell layers thick. And that particular arrangement is diagnostic of the moss family Leucobryaceae. The moss family Leucobryaceae is not found in British Columbia or on Vancouver Island today, but it's quite commonly encountered in the eastern United States and actually most diverse in the tropics. Another morphotype has really anatomically complex midribs. So here's a cross-section through a basal portion of a leaf. Here's a cross-section through a more apical portion of a leaf. In both cases, you can see there's quite a lot going on in that midrib or costa. There's a couple layers of these fiber-like cells, these sort of very circular cells in cross-section. If we look at the cells that make up the rest of the leaf, that's not the midrib, you can see a magnified view of them down here. There's a little tiny bumps called papillae. They're quite low, so they're kind of hard to identify, but here's one just above the pointer, another one right there. The combination of having these little papillae on the leaf cells and that kind of anatomy in the midrib is indicative of the family Podiaceae. And the moss family Podiaceae is one of the most speciose moss families today. They're quite common in really exposed environments like cliff faces and deserts. But you would certainly find extant relatives of this Podiaceous moss on Vancouver Island, especially if you're out looking at like a rock outcrop. There's several other morphotypes that are also have pretty complex midrib anatomies. This is one of them. Again, you see a couple of those bands of fiber-like cells and these very circular cells at the center. But the overall shape of these leaves is totally different from the last morphotype. If we look at a basal section of a leaf, like right here, the leaf bases are really wide and the more apical sections are quite narrow. So this would mean you'd have a leaf with a broad base and a very needle-like distal portion of the leaf. So these would resemble sort of like a very, very fine needle. And the combination of that leaf morphology with that kind of midrib anatomy is diagnostic of the family Dichronaceae and really resembles the extant genus Dicranum. And many of you may recognize this little moss, sort of has a windswept look to it. And the leaves always look like that. This is one of the most common mosses that you'd encounter out on a walk in the forest on Vancouver Island today. So it's kind of cool to see that a relative of this has been on the island for about 80 million years. This is another one of the mosses with a pretty complex midrib anatomy. But what is really unique about this moss, not so much the midrib anatomy, but these little files of cells sticking up from the upper surface of the leaf. Here's one that's fairly well defined. These are called lamellae, and the combination of those lamellae on the surface of the leaf, some unusual shape of the leaf cells, these mammalose cells, suggests affinities with the family Polytrichaceae, and in fact, affinities within a basal grade in that family. And there's actually relatives of this basal grade that live in British Columbia today, like Bartramiopsis lascuriae. So these basal Polytrichaceous mosses have been kicking around in British Columbia for a long time. There's another basal Polytrichaceous moss described from Apple Bay, the locality that Gar Rothwell spoke about earlier, that's quite similar to this morphotype. But the specimens from Apple Bay are quite a bit older. They're early Cretaceous. This locality is late Cretaceous. So it suggests that these basal forms have survived and were quite similar across the Cretaceous on Vancouver Island. There's also a moss which resembles derived Polytrichaceous mosses like Polytrichum. Here's the fossil here, the extant relative on the bottom. You can see that lamellae are much more numerous in this type and they're a little bit taller. The sides of the leaf also kind of enclose the lamellae, making a little chamber as well. And this is typical of the genus Polytrichum. So the whole range of Polytrichaceous mosses, from sort of the most basal members of that group to the most derived members of the group, are actually present in this concretion. This is also a moss you'd pretty commonly encounter out in a walk in the woods or in a meadow or a bog on Vancouver Island today. This morphotype stands out from the others because it has a relatively simple anatomy. The stems don't have much internal differentiation, and the midribs have basically no differentiation. They're all composed of isodiametric cells. This particular arrangement is diagnostic of the moss family Gramiaceae, and there are quite a number of Gramiaceous mosses on Vancouver Island today, especially if you're walking in a creek bed. Many of the mosses covering boulders would be a member of this family. So far, we've just been talking about the Aquacarpus mosses, the mosses that aren't particularly branched, sort of form little cushions. Next up are the Pleurocarpus mosses. These are the mosses that form these really dense mats as epiphytes on maple trees on Vancouver Island today, and also form those beautiful moss mats on the forest floor. It's also worth pointing out that these are the most diverse groups of mosses today. Pleurocarpus mosses account for a very significant portion of moss diversity, but we really don't understand their evolution prior to the Cenozoic very much at all. There are very few pre-Cenozoic Pleurocarpus mosses, so discovery at this locality is really exciting. The first Pleurocarpus Moss morphotype is this one. It has a really unusual feature of having 3 midribs. So here's 1, 2, and 3. It's actually a feature that is unknown from any extant mosses, but it is something that is known from these 2 genera, Tricosta and Crassulaviella, which were described from Apple Bay, that early Cretaceous locality, which I've mentioned earlier. And based on these 2 genera from Apple Bay, an extinct family, the Tricostaceae, was erected. And this extinct Pleurocarpus moss family apparently was around for the entirety of the Cretaceous in Apple Bay, about 136 million years ago. And here, somewhere around 80 million years ago, we have this third type of tricostate moss, which appears pretty different and probably represents a new genus in that family. But this represents a form of Pleurocarpus moss that can recognize as a pleurocarp for various reasons I want to explain here, but we don't have any more. So it's cool to see that extinct diversity. The final morphotype I'll show you is this Hypnalian moss, which really resembles members of the order Hypnales. Originally, I thought this was a leafy liverwort because it has very simple anatomy in the stems. It has very delicate leaves, which almost appear to be just 1 cell thick throughout. But if you look at this, where the leaf is a few cells thick. And this is exactly what we see in members of the moss order Hypnales. So here's a microscope slide showing a whole leaf of the genus Necera, which is and if you look carefully, there is this dark zone. This is that midrib, and it's just a few cells wide and a few cells thick. That's precisely the characteristic we're seeing in this fossil. And this is quite exciting because this is the oldest occurrence of a Hypnalian moss in the fossil record by about 25 million years. And Hypnales is the most speciose moss lineage today. So we can push back our understanding of when this group originated thanks to this fossil. So hopefully I've demonstrated to you this locality greatly enriches our understanding of Cretaceous moss diversity. It's the richest pre-Cenozoic bryophyte flora ever found. And the most exciting thing about it is that it tells us that all major groups of mosses not only had evolved by late Cretaceous, but were quite diverse by then. And that's quite a bit earlier than predicted by molecular dating techniques. I'd like to thank all the folks who put this symposium together and have been out with us in the field to collect fossils, as well as my sources of funding and the Northern Forest Atlas, provided the beautiful extant images of extant mosses I used throughout the talk.
Thank you, Alex.